Welcome to the cover page with Mr. Wilson. Today we're going to be looking at planning and design, which is of course one of those labs that is to be covered by the CSEC Biology syllabus. The topic today is nutrition, and this is of course going to be an example of how to write the lab report. Now this lab usually begins with observation, and the observation is that problem to be solved or that which you have interest in finding out. Here Ben wrapped green mangoes with paper to shorten the time taken to ripe. Now we can't just take this at face value. As scientists, we must do an experiment to see if it is true. Hence we have to design what is called a hypothesis, and the hypothesis is a fixed position taken about the observation that of course can be tested. Here the hypothesis is that wrapping green mangoes with paper shorten the time they take to ripe. Now the hypothesis might not be true, and as such we have to test it, and we use an aim to move forward with the experiment. Now the aim here is to determine if wrapping green mangoes in paper will shorten the time they take to ripe. Now the aim is a clear statement or position for doing the lab. So here we want to find out if wrapping the mangoes will shorten the length of time it takes to ripe. Then we have apparatus and of course material. And apparatus and material is pretty much a list of equipment which could be scientific or otherwise, and the material is pretty much a list of items not including apparatus nor reagent that is used in the experiment. And there we had the apparatus and material on the screen for this particular lab. Then we have here the procedure, and if you observe, the procedure is pretty much a step to achieving the objective. Now this represents a clear series of steps to be taken when carrying out an experiment. It should show how apparatus material will be used in the experiment, and of course it captures things like the amount of time, the measurement for things that will be used in the lab, and pretty much guide to executing the lab. Here we have step by step how we're going to execute this particular lab. It's important to know that for planning and design, unlike the other labs, this lab, the procedure is written in future tense, not past tense as it were for other experiments that we have done for the CSEC Biology. So be reminded that for planning and design, your procedure must provide that clear guideline, and of course should be written in future tense. Now your procedure here or method for planning and design should capture that which is called a control. The control is a setup in an experiment that maintains the manipulated and responding variable. It is used as pretty much a standard in the experiment and may indicate if changes in the experiment is due to manipulation or the theory being tested. Another thing you want to ensure is that in your procedure, the steps that is being tested is repeated just to ensure that you are not exposing the experiment to chance result or what we refer to as confirmational bias. Now, having done that, we need to get to what the expected result is going to be, and we should have an idea if it is true what is going to happen, and if it is not true what's going to happen. Here, if the experiment is true, the wrapped mangoes will ripe before the unwrapped mangoes. So if that happens, then it means that the hypothesis tested is pretty much supported by the findings. However, if that doesn't happen, then you can see your result there on screen. And pretty much it's the same thing or similar thing for all three areas being tested. The expected result is expected to be the same. However, your findings might be different, I don't know. And if that is going to be the case, you have to document it in the experiment. Remember that this is not really a final position. While your experiment might prove that the hypothesis is true, other scientists might decide to continue the research. Now, one thing that you need to look at is what this result can be used for, and here this result could be used to determine or pretty much influence the storage of fruits, and of course the packaging of fruits, and of course the preparation of fruits for the market and pretty much for shipping. Now we get to a point that we refer to as the assumption, and the assumption is just a position taken that something is true without any proof. So we are assuming here that the detergent will destroy any foreign matter that will negatively impact the results of this experiment. Hence, we had washed the fruits to ensure that we are removing things that could influence the outcome of the experiment. Then finally, we have the limitation. And the limitation refers to condition which cannot be controlled in the experiment. And these may, of course, affect the result of the experiment. Here our limitations are the exact maturity of the mangoes is not known. There is also no knowledge of ethylene production and movement in the experiment. You'd understand that ethylene is pretty much that ripening gas, and in some cases it's pretty much coming from the fruit itself, and of course nicely used to get fruits to ripe very quickly. Now, if you would have mastered all that we've gone through with this experiment, then your mark scheme will be 1 mark for the hypothesis, 1 mark for the apparatus material. The method or the procedure could be pretty much divided to give you 5 marks thereabout for the procedure well outlined. The control in the experiment is pretty much 1 mark and the limitations 2 mark. Now, we must make sure that we follow the guidelines for lab. Here you are observing to the left of the lab, there's a space left there for the scientific report or the feedback area where your teacher, your supervisor will be writing their comments. Now, please be reminded that this is an example of the planning and design lab, the planning and design lab report. Now, persons might tweak it somewhat a little bit, but if that is done, never mind, your teacher will always provide the needed guidance. Be reminded, after you would have completed your planning and design for the CSEC Biology, then comes the implementation. Now, the implementation is pretty new, but it's a lab that carries a pretty heavy weighting, so you want to ensure that the planning and design is done properly. It is this that you have written in your planning and design that you are going to be testing in the implementation, so you're going to be carrying out the lab in the implementation. Thanks much for watching, and please be reminded to like, share, and subscribe if you found this helpful, and remember to tell a friend. Until next time, take care.